Good evening, everyone. Today's class is going to be on periodontal microbiology. So in today's class, we'll be seeing about what are the different types of microorganisms, what are the different types of attachments on the tooth, like the plaque, the materia alba, and the calculus, what is the composition of each of them, and how does a plaque form, what are the different types of plaque hypothesis, what is the role of microorganisms in periodontal disease progression, what is the role of biofilm and microbes in dental implant diseases, and what are the strategies to control these oral biofilms. So to move on to the introduction, so the colonization of the oral cavity begins as soon after the time of birth. So within hours after the birth, both facultative and aerobic bacteria will begin to infiltrate into the oral cavity. From the second day after postpartum, anaerobic bacteria will begin to dominate. So after the age of 2 years, there are approximately 10 power 14 microorganisms consisting of 400 different types of bacteria, which are seen in the local cavity. So after the tooth eruption is complete, there are 500 different types of species in the mouth. So this is one MCQ question. There are 500 different types of species in the mouth after the complete tooth eruption has happened. So let us see the basic definitions of plaque, materia alba, and calculus. So plaque is a resilient yellow grayish substance that adheres tenaciously to intraoral hard and soft tissue surfaces. So it's primarily made up of bacteria. So this bacteria is embedded in a matrix of salivary glycoproteins and extracellular polysaccharides. So the plaque is also considered as a biofilm, and it is impossible to remove plaque by using rinses or by sprays, and it is usually done only with the help of routine complete scaling and root cleaning procedures. Next is Materia alba. So Materia alba is a white cheese-like accumulation. The difference between plaque and Materia alba is that Materia alba can be removed using a water spray. Then is calculus. So calculus is a hard deposit that is formed by the mineralization of dental plaque. So calculus, a plaque which is left for a long time, can get mineralized because of the salivary glycoproteins and start forming a calculus. The calculus is always covered by a layer of unmineralized plaque, which can later mineralize again to form a hard layer of calculus. So it is the plaque which is said to be most important for causing the periodontal diseases and the gingival diseases. And apart from that, we also need to know that the microbial composition in the dental plaque is the chief component of periodontal disease progression. So let us understand the composition of dental plaque. So the dental plaque is primarily composed of microorganisms. So these organisms, as I already mentioned, are embedded in an intracellular matrix. So this is an MCQ question. 1 gram of dental plaque contains 10 power 11 microorganisms, especially bacteria. Supragingival plaque, I'm not talking about the whole plaque, but supragingival plaque on a single tooth surface contains more than 10 power 9 times of bacteria. Healthy pocket contains 10 power 3 quantity of bacteria, and a deep pocket has 10 power 8 quantity of bacteria. So there are certain non-bacterial or non-bacterial organisms which are also found in the plaque, which are mainly the mycoplasma organisms, the yeast, the protozoa, and viruses such as the cytomegalovirus and the Epstein-Barr virus. Now, we have to classify plaque mainly to understand the differences and the composition of each type of plaque. Plaque can be broadly classified into supragingival and subgingival plaque. So, as the name suggests, supragingival is the plaque accumulation above the gingival margin, whereas subgingival is the plaque that is located below the level of gingival margin. So the supragingival plaque can be further classified into coronal plaque and marginal plaque. So marginal plaque is the portion of the supragingival plaque that is located near the marginal gingiva, and the coronal plaque is the plaque that is located coronal to the marginal plaque. The subgingival plaque, as I already told you, is the plaque that is located below the level of the gingival margin. That is of two different types. One is tooth associated and one is tissue associated. Tooth associated plaque is found in association with the root surfaces, and tissue associated is found in association with the soft tissue wall of the periodontal pocket. This is also an MCQ question. Moving on to the supragingival plaque, as I already mentioned, it is located above the gingival margin. It demonstrates a stratified organization of a multi-layer accumulation of different types of bacteria. So it is stratified. So at the tooth surface, it is mainly made up of gram-positive cocci and short rods. And on the outer surface, it is mainly made up of gram-negative rods, filaments, and spirochetes. This is the composition of supragingival plaque. So the coronal plaque is already mentioned is contact only with the tooth surface, whereas the marginal plaque is seen along the gingival margin. Next, moving on to the subgingival plaque. So subgingival plaque is located below the gingival margin and it is located between the tooth and the gingival pocket epithelium. So since the subgingival plaque is completely cut off from the oral cavity, it derives the nutrients from the local availability of the blood product which is located inside the gingival pocket. So the host inflammatory cells and the inflammatory mediators will have a great influence on the establishment and the growth of bacteria in the subgingival region. So the composition will depend upon the probing pocket depth. So as the probing pocket depth keeps increasing, the condition is going to become more anaerobic. Hence, more anaerobic bacteria will begin to dominate. So the apical part is usually dominated by the spirochetes, the anaerobic cocci and the rods, whereas the coronal part is mainly dominated by the rods and the filaments. Okay, so this slide is very important because it will basically summarize the difference between the supragingival and the subgingival components. The supragingival plaque is mainly made up of gram-positive and gram-negative organisms, whereas the subgingival plaque is mainly made up of gram-negative organisms. Coming to the morphology, the supragingival plaque is mainly made up of cocci, branching rods, filaments, as well as spirochetes, whereas the subgingival plaque is dominated by the rods and the spirochetes. Depending upon the aerobicity, supragingival plaque can be facultative with some anaerobes, whereas subgingival plaque is strictly anaerobic in nature. Depending upon the reaction of the host, supragingival plaque can cause both dental caries as well as gingivitis, whereas subgingival plaque is mainly responsible for the formation of gingival disease as well as for periodontitis progression. Depending upon the motility, in supragingival plaque, it is firmly adherent to the plaque matrix, whereas in subgingival plaque, it is less adherent because it has more motile forms of the bacteria. Now, moving on to the next topic, which is the selected bacterial species that are found in the dental plaque. If you can remember this slide by heart, it will answer all the questions, mainly because it will tell you what are the gram-positive organisms, what is the nature of its anaerobicity, whether it's aerobic, anaerobic, facultative, or strictly obligate anaerobes. So the gram-positive facultative anaerobes, which are found in the plaque bacteria, are Streptococcus mutans, Streptococcus sanguis, and Actinomyces viscus. The gram-negative facultative bacteria which are present are Aggregatibacter, Actinomycetum comitans, Capnocytophaga species, and Econilla corrodens. Gram-negative anaerobic bacteria are Porphyromonas gingivalis, Provatella intermedia, Tandrella forsythia, Fusobacterium nucleobatum, and Campylobacter rectus. And finally, we also have spirochetes such as Treponema denticola. As we come in the forthcoming slides, you will note that these organisms are grouped into colored complexes, which is given by Socranski. That I will be discussing in the further slides. Okay. Now we are going to be learning a basic detail with exam importance on regarding the dental plaque formation. So in dental plaque formation, we mainly need to understand this mechanism because this will be able to judge regarding the type of composition at every stage of the plaque formation. The first stage of dental plaque formation is the formation of the dental pellicle on the tooth surface. Next is the initial adhesion and attachment of the bacteria. And the third step is the colonization and the plaque maturation. So please remember these three steps because it is of important exam relevance. The first step is the formation of the dental pellicle on the tooth surface. The second step is the initial attachment and the adherence of the bacteria. And the third step is the colonization and the plaque maturation. So the dental plaque may be readily visualized on the tooth surface if no oral hygiene is performed for 1 to 2 days at maximum. Okay, so moving on to the first step, which is the formation of the pellicle. Within nanoseconds after vigorously brushing and polishing the tooth surface, a very thin saliva-derived layer called the acquired pellicle will form on the tooth surface. The acquired pellicle is the starting point for plaque formation. Within the first 24 hours, the pellicle is about 0.1 to 0.8 micrometer in diameter. This is again an MCQ question. 24 hours, pellicle thickness is 0.1 to 0.8 micrometer in diameter. So derived from the components of the saliva as well as the gingival clavicular fluid, pellicle can also be got from the bacterial cells themselves and also from the host tissue cells and the food debris that is accumulated in the intraoral cavity. So with that, the pellicle is formed. Next is how does the bacteria adhere and attach to the tooth surface? That happens in 4 different stages. The first stage is the transportation of the microorganism to the tooth surface. Phase 2 is the adhesion of the bacteria to the tooth surface. Phase 3 is forming a strong attachment on the tooth surface. And phase 4 is the colonization on the surface with maturation of the biofilm that is being formed. We will be seeing in brief about each phase now. So phase 1 is how the bacteria gets transported to the surface. So initial transportation of the bacteria to the tooth surface will occur because of 4 different mechanisms or random contacts. The first mechanism is called as Brownian movement. Brownian movement is when the bacteria moves in random directions in zigzag motions, and it will be able to go and attach itself to a tooth surface. And the average displacement speed is about 40 micrometer per hour. Next is sedimentation of the microorganisms, either from the saliva or from the GCF subgingivally. Third is the liquid flow. If the salivary flow is said to be diminished, then the surface bacteria will not be able to wipe away from the tooth surface. Hence, it will adhere to the surface of the tooth. And the final is mainly by active bacterial movement or chemotactic activity of certain bacteria. So this is the mechanism of transport. Next, how is the initial adhesion of the bacteria that is occurring? So this is the initial reversible adhesion. Please remember, the initial adhesion is said to be reversible. That is, we will be able to remove it without any much of an effort, like using a water spray. So it is initiated by the interaction between the bacteria and the surface energy on the tooth from a certain distance of less than 50 nanometers. So only if the bacteria is in 50 nanometer distance, it will be able to associate an interaction. And this will happen through long ranges as well as short range forces such as van der Waals forces and electrostatic repulsive forces. So during this stage, there are 2 different types of forces. One is the van der Waals forces and next is the electrostatic repulsive forces. So the initial attachment has also occurred. So how to get a permanent attachment? That is the phase 3 of the formation. So after the initial adhesion, a firm anchorage will occur between the bacteria and the tooth surface, which will be established by other types of interactions such as covalent bonding, ionic bonding, or by hydrogen bonding. Please remember, the previous one was electrostatic forces or van der Waals forces, whereas this forces is mainly covalent, ionic, and hydrogen bonding forces, which are more permanent in nature. So bacteria are better protected by means of making it from a reversible bonding to an irreversible bonding nature. So the bonding between the bacteria and the pellicle on the tooth surface is mediated by the extracellular proteinaceous components also. So apart from these forces, the extracellular matrix proteins such as adhesins will also help to form a strong attachment by the presence of receptors present on the tooth surface and the receptors which are present on the organism. So once the attachment of the bacteria to the tooth surface is completed, it is known as the first step and the most crucial step of any biofilm or plaque formation. So many bacterial species will contain certain surface structures such as fimbriae and fimbrials, which will help in the attachment to the different tooth surfaces. So with this, we have seen how the bacteria is firmly attached to the tooth surface. And now we will be seeing about the colonization and the plaque maturation. This is what is the most important from exam point of view. So I am explaining this in detail. The primary colonizers, as the name suggests, the primary colonizers are the bacteria which will initially come and invade or attach to the tooth surface. The primary colonizers, also called as early colonizers, are the Streptococcus species and the Actinomyces species. I am repeating, the primary colonizers are the Streptococcus species and the Actinomyces species, which use the oxygen and which will lower the redox potential of the environment, which will favor the growth of anaerobic species. So how they do this is, they will go attached to the tooth surface, reduces the redox potential and will allow the growth of other anaerobic species. Next, the secondary colonizers, that is the colonizers which come after the primary colonizers, they do not initially colonize the surface of a clean tooth. That is, secondary colonizers are Prevotella intermedia, Prevotella loci, Capnocytophaga species, Fusobacterium nucleatum, and Porphyromonas gingivalis. So once the secondary colonizers also come into play, now we have to understand how the primary and the secondary colonizers interact. For this process is called as co-aggregation. So the ability of 2 genetically different bacteria to recognize each other and to adhere to one another is called as co-aggregation. So we saw how the bacteria initially attached to the tooth surface. Now we have to understand how the genetically different bacteria attach to each other, right? So this process is called as co-aggregation. So the ability of 2 genetically different bacteria to recognize and to adhere to one another is called as co-aggregation. This co-aggregation is based on specific interactions on the surface of the proteinaceous bacteria on one bacteria with the respective receptor, which is usually a carbohydrate or a protein receptor, that is present on the other bacteria. So the adhesions, that is the receptors which play an important role in adhesion, are of various molecular sizes and they compete with each other for binding to the receptor polysaccharide. So to one organism, more than two organisms can come and bind to. So this co-aggregation is a very important process in the plaque maturation and for communication between the different types of bacteria. So we will see how the co-aggregation happens. Secondary colonizers, as I already mentioned, they will not be able to bind into a clean tooth surface because you need the primary colonizers to come and reduce the redox potential. So well-characterized interactions between the primary and the secondary colonizers are, please remember these three, they are also of exam importance. One is Fusobacterium nucleatum, which will go and bind to Streptococcus sanguis, which is a primary colonizer. Protella intermedia, which will come and bind to Actinomyces viscus, which is also a primary colonizer. Then we have the Capnocytophaga species, which will also come and bind to Actinomyces convenans, which is a primary colonizer. Streptococcus will show intrageneric co-aggregation, which means Streptococcus species will be able to bind within one another itself. This is called as intrageneric co-aggregation, allowing them to bind with the nascent monolayer of the already bound streptococci. In the later stages, co-aggregation will happen between 2 gram-negative bacteria also, like Fusobacterium nucleatum, P. gingivalis, or Treponema denticola. So please remember the organism which co-aggregate with one another. It is the Fusobacterium with Streptococcus sanguis, Protella with the Actinomyces species, and the Capnocytophaga also with the Actinomyces species. So one more important point that I would like to emphasize at this stage is that Fusobacterium nucleatum is also called as the bridge species. That is, it will be able to bridge the primary colonizers along with the secondary colonizers. Okay, so these are the examples of co-aggregation. Streptococci going and binding to Actinomyces species, it gives a concave appearance. So in concave appearance, there is usually a central layer of gram-positive organism surrounding a layer of gram-negative organism. And in test tube appearance, the filamentous bacteria will begin to adhere to gram-negative rods. Okay, so with this, we have seen completely in detail about dental plaque. What are the different types of dental plaque? Supragingival, subgingival plaque. What is the composition of supragingival and subgingival plaque? What is the steps of dental plaque formation and what are the organisms and how they come in adhere to the dental plaque? Now we are moving on to the next phase of the class, which is the Socranski's microbial complexes. So the periodontogenic bacteria, or the not periodontogenic, generally periobacteria, are divided into 5 different complexes by Socranski. So Socranski et al. In 1998 examined over 13,000 subgingival plaque samples from 185 adult subjects and did DNA hybridization technology to describe the presence of specific complexes of periodontal microorganisms. So, if a question is going to be asked as to who gave the microbial complexes in periodontal microbiology, it is Socranski. And how did he classify them? It is based on the role that they play in the periodontal health and periodontal disease progression. Please remember, the microbial complexes are classified based on the role of the bacteria and the group in periodontal health and periodontal disease progression. So, depending upon this classification, he is given 5 complexes of bacteria. In that, the primary colonizers are the yellow complex and the purple complex. The yellow complex comprising of the Streptococcus species and the purple complex comprising of the Actinomyces species. The secondary complex are 3. You have the green complex, the orange complex, and the red complex. The green complex is made up of Iconilla corrodens, Actinomyces comitans, the Capnocytophaga species. The orange complex is made up of Campylobacter species, the Protella intermedia, and Fusobacterium. And the red complex is mainly comprised of P. gingivalis, Tannerella forsythia, and Treponema denticola. You remember the previous slide where I mentioned that the colonizers will be grouped according to its function. You can see that the primary colonizers are predominantly yellow and green complexes, whereas the secondary colonizers are usually green, orange, and red complex bacteria. Some of these bacteria are part of the regular flora of the mouth and others are acquired from the mother to son or by horizontal transmission. The periodontal pocket contains a very fertile ground for the growth of many organisms. That is why you will be able to see a lot of gram-negative anaerobic supply in these regions. This means that the organism will be able to proliferate and thrive in an oxygen-free environment like a periodontal pocket. Each bacteria taken individually cannot be pathogenic as much as they form when a complex is being formed. The categories are based upon the pathogenicity of the bacteria and their role in the development of plaque. This is what I mentioned of MCQ importance. The categories are based on the pathogenicity of the bacteria and the role that they play in the development of the dental plug. So we'll be just seeing in brief about each. The yellow complex has not been directly associated with changes in the clinical signs or periodontal diseases. The purple complex are the first ones to colonize in the subgingival area, and it has also not been found to associate with gingivitis or periodontal conditions. It seems to pave the way for the other bacterial components to come into action. It is frequently found in combination with the actinomyces species. The green complex. The green complex bacteria, which is mainly the aggregative bacteria actinomycetum competens, is usually found in association with a pathologic periodontal pocket. But it is also not found in severe stages of periodontal disease progression. Next, we have the orange complex bacteria. Orange complex bacteria are always closely associated with one another present in the group and also related to the red complex bacteria, and hence the orange complex and the red complex are mainly responsible for periodontal disease formation and progression. Apart from that, it is also associated with increased probing pocket depth and increased chances of bleeding on probing. Finally, the red complex bacteria. The red complex bacteria, mainly the P. gingivalis, Supra denticola, and Tandrella forsythia, are the individual species that are responsible with increased bleeding on probing, severe forms of periodontal disease also. So this red complex bacteria establishes a very strong relationship with the probing pocket depth and also has an increasing tendency for more bleeding on probing and increasing clinical parameters. Sites which harbor P. gingivalis alone, which is a red complex bacteria, or in combination with the other two species, that is Treponema denticola and Tandrella forsythia, are seeming to have the deepest periodontal pockets within the oral cavity itself. So, these bacteria will have certain metabolic interactions between them. How will the bacteria exist alone? So, it has to have some interaction and support from the other bacteria, right? So, this is called as metabolic interaction among different bacterial species. So, this is called as synergism. That is, the presence of one bacteria is going to help the growth of the other bacterial species. Lactate and formate, which is byproducts of the metabolism of streptococci and actinomyces, can be used as a metabolic food intake for the other plaque microorganisms. This is also a form of synergism. So when a byproduct, that is the excretory product of one bacteria, is used as a source of energy by the other bacteria. So this concept is known as synergism. So the growth of P. gingivalis is also enhanced by the metabolic byproducts from other species of other microorganisms such as succinate, which is got from Capnocytophaga, protoheme, which is also got from Campylobacter rectus. So please remember these two examples. P. gingivalis is taking the byproducts of Capnocytophaga, which is a succinate compound, and also takes a protoheme, which is secreted by the Campylobacter rectus as a byproduct. Okay. So now let's move on to the microbial specificity of periodontal diseases. So since time immemorial, many authors have been trying to understand the role of these bacteria and microbes in periodontal disease progression. So there were 4 different types of hypothesis which was given over the period of time. So the first one is called as the non-specific plaque hypothesis, which was given by Loche in 1976. The author name is very important for this hypothesis because hypothesis and author names are frequently asked questions in PG exam. So in the mid-1900s, periodontal disease was believed to result from an accumulation of plaque over time with a diminished host response and increased susceptibility with age. So non-specific plaque hypothesis says it's the quantity of the plaque which plays an important role and not the quality of the plaque. So it says that periodontal disease results from an elaboration of noxious products by the entire plaque flora, and this led to a concept that when you control for the control of the periodontal disease, we need to control the amount of plaque accumulation. According to this theory, all plaque are considered equally pathogenic. Please understand, non-specific plaque hypothesis says that it is not the quality of the plaque. It doesn't talk about the composition of the plaque, but it says it is the amount of plaque that has formed which causes the nature of the disease progress. So during this non-specific plaque hypothesis era, it was thought that the main aim of treatment was to remove the plaque by non-surgical or surgical mechanisms. But it is only after this that people began to understand there are certain people who have little plaque accumulation, but they have exaggerated disease. And there are certain people who have less plaque accumulation, exaggerated disease, and some people who have less plaque, but more exaggerated disease. So they thought that it is not the quantity of plaque that matters, but it is the composition of plaque that matters. Then came the specific plaque hypothesis, which was given by Newman Socranski in 1977. So this states that only certain plaque is said to be pathogenic, and this pathogenicity depends upon the presence or increase of certain specific microorganisms. Plaque harboring specific bacterial products or specific bacterial pathogens results in different types of periodontal disease. For example, it is only now that they realized in 1977 increased levels of actinomycetum aggregatibacter actinomycetum comitans is found in localized aggressive periodontitis. So this was the next specific plaque hypothesis which was given. Next, in 1994, Marsh and colleagues gave the ecological plaque hypothesis. So what they said is, it is not only the composition of plaque, but also the environmental factors which play a very important role in the disease progression. So change in the key environmental factor will trigger a shift from the balance of the resident plaque flora that may predispose to the formation of the disease at that particular site. So this hypothesis is based on the theory that there is a unique microenvironment present in each plaque composition in the local area, which will influence the type of organism that is present there. So it was also introduced that the disease can be prevented not only by inhibiting or preventing the plaque accumulation, but we'll also have to interfere with the environmental factors, which will help in the growth of certain bacteria also. Okay, so this is the latest theory which has been given, which is called as the Keystone pathogen hypothesis, which was given by Haji Shengalis in 2012. So he said that there are certain low abundance microorganisms which, even when present in lower numbers, will be able to trigger an inflammatory and an immune response that can cause a disease progression. So he said that most of these microorganisms are in the red complex bacteria, and it is P. gingivalis, that is Porphyromonas gingivalis, which is considered as the keystone pathogen. So keystone pathogen is one pathogen which even when present in lower numbers or in subclinical numbers will be able to still produce a disease manifestation. So this P. gingivalis will act by three different mechanisms. The first mechanism is that it will interfere or destroy the complement system. It will cause increased activation of interleukin 8, thereby increasing the inflammatory process, and will also manipulate the TLR receptors. I hope you remember the previous class. In TLR, there is a toll-like receptor. They are seen as the gate control receptors. They will only trigger the inflammatory process. So this P. gingivalis will go and crosstalk. That is, it will confuse the TLR receptors, thereby causing an immune malregulation, thereby increasing the inflammatory diseases. Okay. So what was the criteria for identification of this periodontal pathogens? As I already told you, there are 500 different types of species in the oral cavity. So how do we know whether an organism is causing the disease? This is a very, very important repeated MCQ question. This will also come in microbiology. So how to identify the organism responsible for a disease? It is based on Koch's postulate, which is given in 1870. So this is based on 4 different rules. The first rule is that the pathogen should be routinely isolated from the deceased individual. It should be grown in a pure culture in a laboratory. It should produce a similar disease when the pathogen is inoculated into another lab animal, and they should also be able to recover from the lesions in the diseased animal also. So these are the 4 important Koch postulates which you will have to by heart. So Socranski in 1992, he modified this Koch postulates in order to fit it for the periodontal microera. So he said that the microorganism must be associated with the disease, and an increase in the level of microorganism should be seen in the diseased site. The microorganism must be eliminated or decreased in sites which have undergone treatment. They must be able to demonstrate a host response if the microorganism is inoculated. It should be able to cause the same disease pattern in an experimental animal model, and they should also demonstrate virulence factors in order to cause the disease progression also. So 5 important factors as given by Sigmund Socranski in 1992 is the microorganism should be associated with the disease. It should be decreased after treatment. It should show visible host response. It should be able to cause the disease in a lab animal, and it should also demonstrate virulence factors at the time of disease progression. So how does these microorganisms work? So for the bacteria to work, it should demonstrate certain potential pathogenic features. So it has to be able to colonize a subgingival area, and it should produce factors that can either directly or indirectly damage the host tissue. So in order to colonize the subgingival sites, the species can do it in the following ways. Either it directly attaches to the tooth surface, or it co-aggregates with the already existing microorganism. It multiplies very fast. It has a favorable interbacterial relationship. It should be able to overcome the host defense mechanisms. It should help in host tissue damage. It should invade into the host tissue, or it should produce factors that will destroy the host tissue, or it should have virulence factors that will predetermine the genomic era in the local site. So I'm just briefing you about the various virulence factors that are present. I am not going in detail with each organism because it has to be by-hearted. It is given as a table in the Pulse textbook. Please by heart the table. It will tell you the different virulence factors of very important periodontogenic bacteria, which you can by heart by yourself. So I'm just going to brief about what are virulence factors. So virulence factors are those factors which when present in an organism will be able to increase the pathogenicity of the organism. So virulence factors can be subdivided into factors which will help in colonization, toxins and enzymes that will degrade the host tissues, and mechanisms that will protect the pathogenic bacteria from the host. So what are the other two factors? Other factors is in order to colonize the pocket, the bacteria must adhere to cells or tissues in the region such as the teeth or the pocket epithelium or by co-aggregation with the specific bacterial biofilm. So the bacterial cell structures which provide these functions are the fimbriae or the pili. So the P. gingivalis has two fimbriae which are known as the major and the minor fimbriae. And Actinomycetum comitans has pili and also carbohydrates which are present on its cell surface, which will help to go and attach to the tooth surface directly. Treponema denticola has the major sheath protein, which when it interacts with the host extracellular matrix such as fibrinogen, fibronectin, and laminin. P. gingivalis and Fusobacterium nucleatum also contain major surface proteins, which are also promoting in order to bind the host and the bacterial cell surface receptors. Next is factors that promote the host tissue destruction. So there are many factors that are produced by the bacteria which will directly go and destroy the host. It is the fimbriae of the P. gingivalis and aggregative bacter are said to be highly antigenic in nature. So inflammation is also a major contributor for host tissue destruction in periodontal diseases. So the inflammation which is triggered by the bacteria itself will destroy the host tissue. Bacterial proteolytic enzymes such as tryptin-like proteases which are produced with the red complex bacteria will also increase the periodontal disease progression. For example, in P. gingivalis, there are enzymes called as gingipains. Gingipains are serine proteinases which are responsible for the degradation of the surface proteins and for degradation of the host tissue components. So gingipains are multifunctional proteins and they play a very important role in adhesion, tissue degradation, as well as the evasion of the host responses. Next is how do they strategize and give an entry into the host immunity. So the bacteria will be able to produce extracellular capsules like case of P. gingivalis. It can cause proteolytic degradation of the host immune cells. It can modulate the host response or it can directly invade into the gingival epithelial cells. These are the strategies for evading into the host immunity. Here I have just briefed about aggregative bacter, Actinomycetum comitans and P. gingivalis. These are two very important periopathogenic bacteria. Whatever I have given in these two slides are more than enough to appear for your NEET MCQ exams. So aggregative bacter Actinomycetum comitans was identified in 1912 by Klinger. So Klinger is an MCQ. It was also named because of its association with Actinomyces israeli in actinomycotic infections. The genus name Actinomycebacillus refers to actins means star-shaped rod, that is bacilli. There are 6 serotypes of aggregative bacter A, B, C, D, E, and F. Out of that, A and B are found in the oral cavity. MCQ question again. Serotype A and serotype B are located in the oral cavity, whereas serotype C is seen in extraoral infection. That is not asked in an MCQ, but A and B intraoral is an important MCQ question. Serotype B is found in association with localized aggressive periodontitis, and serotype A is seen in chronic periodontitis. All the points that are given here are MCQ questions. They are gram-negative, approximately 0.4 into 1 micrometer in size. They are non-motile, they are sacrolytic, capnophilic, round-ended, facultative, anaerobe. Please by heart this line. It answers almost 10 MCQ questions. Gram-negative, non-motile, sacrolytic, capnophilic, round-ended, facultative, anaerobe. Okay. The virulence factors of aggregative bacter are adhesins. As I already told you, adhesins means it will directly go and bind to the surface of the host by the means of fimbriae, extracellular matrix materials, and extracellular vesicles. You also have invasins, which are mechanisms or craters that are present on the cell surface, which will help in go and attaching itself to the host tissue. It also contains bacteriocins, which will be able to degrade and change the ecological pressure in the local site, thereby preventing the adherence of other organisms. It also has some amount of antibiotic resistance. It also releases leukotoxin. The key characteristic feature of aggregative bacteria is the presence of leukotoxin, which will be able to degrade the immune cells. Apart from that, it also has lipopolysaccharides for adhesion. It also has certain chemotactic inhibitors. It will also has a cytolethal distenting toxin, which means it will have a cytotoxic effect on the neutrophils. It also has immunosuppressive proteins and Fc binding proteins. Fc binding proteins means Fc is a portion of the antibody. So this Fc binding protein will go and attach itself to the antibody and prevent its function in the humoral immune system. Next is P. gingivalis. The three things that you have to know is it's a red complex bacteria. You already know that point. It's a keystone pathogen. I just mentioned in the keystone pathogenesis. It is a gram-negative anaerobic asaccharolytic black pigmented bacteria and is a very potential periopathogenic organism. The virulence factors, as already mentioned, invasion and colonization is mainly via the fimbriae, the hemagglutinin and the vesicles. The evading systems is via the immunoglobulin, the complement proteases, the LPS and other antipathogenic bacteria product which has been released by the bacteria. Multiplication is mainly with the help of proteinases. Proteinases are protein degrading enzymes in the presence of hemolysis, which are hemoglobin degrading enzymes. And also it is involved in the host tissue damage by uses of gingipains. Remember, I told you gingipains are proteinases that will help in degrading into the host tissue. Apart from that, it also has collagenases. It also has trypsin-like activity, fibrinolytic activity, and other keratinolytic activity also. Okay, this is a summary to explain to you how important it is to understand the shift of the microorganism from the health state to a diseased state. So, microbial shift during disease. If you remember this slide, you'll be able to answer many of the questions. When there's a shift from health to disease, the gram-positive organism will become a gram-negative state. That is, gram-positive organisms will become less dominant and gram-negative organisms will become more dominant. It will become from cocci to rods. Non-metal organisms tend to become more motile organism infiltration. Facultative anaerobes tend to become obligate anaerobes. And fermenting bacteria become proteolytic species. So please remember all this. So I have given you a summary of the organisms which are involved in every form of periodontal disease. Just by heart this so that it will be easy for the MCQ. In periodontal health, it is usually the gram-positive facultative species. In gingivitis, it is usually the P. gingivalis. So this was demonstrated by a study done by Harold Lowe and co-workers. That is not very important. And in chronic periodontitis, it is P. gingivalis, pre-intermediate Andrella forsythia, Fusobacterium nucleatum, and Treponema species. And viruses are also involved in this process, and the Epstein-Barr virus and the cytomegalovirus. So chronic periodontitis is 90% anaerobic infection. Localized aggressive periodontitis is associated with aggregative bacter Actinomycetum comitans only. Okay. And the other bacteria which are found are P. gingivalis, C. rectus, and F. nucleatum. And it is a gram-negative capnophilic and anaerobic rod which are mainly found in the localized aggressive periodontitis. In generalized aggressive periodontitis, Papillomavirus gingivalis will dominate more than aggregative bacter Actinomycetum comitans. In necrotizing periodontal diseases like that is necrotizing ulcerative gingivitis and periodontitis, it is usually the P. intermedia and the spirochetes which play a very important role. And in the abscesses of the periodontium, you have the F. nucleatum, P. intermedia, Porphyromonas gingivalis, and Andrella forsythia. Please by heart, I'm just giving you the name of the disease and the microorganism associated with the disease. Okay, so now the latest trend, implants. So what are the biofilms around dental implants? So osseointegration is defined as the direct bone to implant contact without imposition of any other tissue. So longitudinal studies have shown that there are periodontal pathogens which also play an important role in the peri-implant diseases also. So the peri-implant inflammation in the subgingival region is seen as a major contributor for the further implant loss. So peri-implantitis. Inflammation of the peri-implant mucosa which progresses to the destruction of the alveolar bone in contact with the implant thread is called as peri-implantitis. So like how you have a healthy periodontal pocket, a healthy peri-implant pocket will have a high cocci cell and a low ratio for the anaerobic to the aerobic species. And a failing implant, this is the MCQ question which was asked in 2019, a failing implant will have Actinomycetum comitans, aggregative bacter Actinomycetum comitans, P. gingivalis, Andrella forsythia, Fusobacterium nucleatum, and Capnocytophaga. As you can see, there is no much of difference between periodontitis and peri-implantitis. So the organisms is also almost similar in both the conditions. So how does a biofilm form in a dental implant? We saw how it forms on a tooth surface, right? But there is a difference in a dental implant also. So biofilm formation and dental on dental implants and teeth will also form a similar pattern of microbial colonization. Biofilm formation around natural teeth occurs in minutes, whereas in case of implants, it takes as to about 2 to 6 hours to form the initial pellicle formation. This is because the clean tooth surface will have remnants of the unattached microbiota that can immediately multiply and form a favorable position for the other bacteria to come and attach, whereas the surface of an implant will lack the indigenous microbiota. This is the difference between the tooth and the implant surface. So after the exposure into the oral cavity, an implant will form an acquired pellicle around it. So the bacterial attachment with the initial colonizers followed by cell-to-cell adhesion with secondary colonizers will begin to occur at this stage. So this pellicle is derived from components in the saliva as well as components from the GCF also. And it also derives it from the host tissue components and from the other bacterial components. This will act as a substrate which occurs within the early 30 minutes after the implant exposure into the oral cavity. So once this primary substrate is formed, it is a similar mechanism out as to how the dental pellicle forms in the normal tooth surface also. So with this, we have completed the periodontal microbiology class. We have seen how a plaque forms, what is Materia Alba, what is calculus, what are the different types of plaque, what are the different organisms present in the plaque, what is the methods, what are the steps in plaque formation, what are the microorganisms dominating in each step, what are the Socranski's complexes, what are the plaque hypotheses, the 4 different plaque hypotheses. We saw what are virulence factors, what are the different virulence factors in AA and PG. Then we saw the peri-implant plaque composition and how a plaque forms in peri-implant conditions and microorganisms dominating in peri-implant diseases. So I have just mentioned few MCQ questions. Which of the following is least likely to be seen in plaque? It is the Streptococcus sanguis species, which is most commonly associated with the healthy plaque, periodontal sulcus. Aggregative bacter actinomycetum comitans is commonly associated. I just mentioned it is the localized aggressive periodontitis. But in this question, they've given you juvenile periodontitis, adult periodontitis, and peri-implantitis. Aggressive periodontitis was initially called as juvenile periodontitis. So the answer is juvenile periodontitis. A central gram of negative core supporting an outer coccal cells are called as the concave appearance. We just mentioned during plaque formation, right? So central gram-negative core and outer coccal cell periphery is called as the concave appearance. The breakdown of PDL fibers in periodontitis is mainly because of collagenase enzyme, because the PDL fiber is mainly made up of type 1 collagen. So any bacteria which produces collagenase enzyme will be able to degrade the PDL fibers. So with this, we are concluding the class. Thank you very much.